Hello, everyone, and thank you for coming to my presentation. Valerie asked me to talk about seed longevity, and to do this, I'd like to focus on the conditions for seed storage. There are two main factors, the moisture and the temperature, and there are international standards to guide gene banks. However, this is a conference on exceptional species. The narrowly defined standards don't usually work for these species. In fact, when you're in the diversity business, it may be unrealistic to expect the same outcome to standardized treatments. The image of dripping nail polish may seem a bit strange, but it serves as an analogy for the approach that we're taking. Plot spoiler, I'm going to talk about fluids turning to solids. So have an image of hardening nail polish to get through the gist of my talk. We knew way back when that moisture and temperature control longevity. Harrington's thumb rules tell us that you can double longevity for every 1% reduction in seawater content or 10 degrees Fahrenheit reduction in temperature. The viability equations developed by Richard Ellis and Eric Roberts are based on similar principles and provide more guidance. In the first panel, you can see the exponential relationship of longevity with water content. The second panel depicts the modeled relationship with temperature. You guessed it. From the moisture relationship, there was a general conclusion of the drier, the better. It was those model relationships that defined the terms orthodox and recalcitrant. Orthodox seeds followed the models, and recalcitrant seeds didn't. Many of us tested the models and learned that drier wasn't always better. You can't achieve infinite lifespan by drying seeds indefinitely. There's a critical water content, and below which longevity models failed. Too much drying could even decrease lifespans. Drying recommendations for seed storage put seeds at the cusp between no benefit and potential damage, and also gave optimistic estimates for longevity in some circumstances. The effects of temperature were modeled by the viability equations using a quadratic equation with a maximum at minus 40 degrees. That runs counter to cryobiologists' advice to store at liquid nitrogen temperatures to prolong lifespans. The quadratic function wasn't supported by the data. In one case, John Dickey and colleagues developed a logarithmic relationship. At NLGRP, we used Phil Stanwood's liquid nitrogen experiments to estimate longevity at minus 80 and liquid nitrogen vapor, as well as warmer temperatures. But there always seem to be this sort of diminishing returns when we're storing at extremely low temperatures. Research on a broader array of species also revealed that many seeds didn't follow the orthodox pattern of exponential and logarithmic effects of lowering moisture and temperature, respectively. Not in the obvious way like recalcitrant seeds, which died almost immediately. The effects were subtle. Like you could dry the seeds appropriately, but putting them in the freezer was lethal or tended to make them age faster. For lack of a better category, these seeds were termed intermediate. Some people in the seed community interpreted this category as seeds with desiccation tolerance, intermediate between orthodox and recalcitrant. It's possible. Not having a quantitative measure for desiccation tolerance makes this definition difficult to understand and to measure, especially because we can also argue that orthodox seeds have limited desiccation tolerance by virtue of the critical water contents that I showed previously. Regardless, we need better guidance on how to dry this group of seeds to achieve maximum longevity. What about this temperature anomaly? Seeds age faster in the freezer than in the fridge. Does this imply that cryostorage is not a viable option? No pun intended. Longevity models don't help us at these questions, and so we need a different way to look and to think about storage rather than the empirically based models. Engineers deal with the problem of components with different thermal properties working together through broad temperature ranges. So this direction is how we're approaching the problem. Some germplasm are intrinsically pore keepers. Danny Ballesteros and Valerie Pence reported the short lifespans of fern spores and salix seeds. And we see the same things with some pollens. The effect of temperature and moisture follow similar patterns as orthodox seeds. Everything just happens faster. For example, pollen might survive 10 days at room temperature and about a year in the freezer. In all cases, the freezer storage just doesn't buy enough time for gene banks. Adding to the complexity is seed dormancy. Baskin and Baskin tell us that post-harvest dormancy can be released or induced by moisture temperature treatments, and this can affect the interpretation of viability data. So what I'd like to do is show you some of our data on papaya seeds to further explore the intermediate seed condition and to reveal some of our storage options. Papaya seeds are one of the original seeds diagnosed as intermediate, and it's a nice steady system because seeds are readily available from the local grocery store. And there is a broad genetic base if fruits are sourced from Hawaii, Mexico, Guatemala, and Brazil. Here's a typical drying experiment. We expose seeds to relative humidities ranging from 1 to 92%, indicated at the top of the graph, and achieved water contents that are indicated on the axis. Water contents are lower than you might expect because papaya has a lot of lipid. In the short exposure time, less than 3 months, there isn't a lot of effect for seeds held at the 10 to 85% relative humidity in that region. That's not a surprise. This is like a typical desiccation tolerant seed. Aberrant behavior is only observed when you remove all the water or nearly all the water. Looking at the lowest moisture treatments, you can see papaya seeds died quickly when over-dried at a rate that is faster than what I've observed with orthodox seeds. Still, we can conclude that these seeds tolerate a lot of drying, but not tolerate complete drying. Here is some temperature exposure data for papaya seeds. Wait, it's not exposure data, it's storage data. And that's because a brief exposure doesn't reveal anything. It takes time to see these trends. In this case, observations made over a 15-year period. Notice the bar colors reflect storage time of 6, 10, and 15 years. Are you getting the pattern here? It takes time to observe detrimental effects of low moisture and temperature. You don't see these effects immediately like you might with recalcitrant seeds. We stored seeds at a range of temperatures, 5, minus 18, minus 80, and liquid nitrogen vapor. Looking at the 5-degree storage data, that is storage in the refrigerator, you can see that germination is noticeably reduced after 6 years, and all the seeds are dead in 15 years. This is typical of a relatively short-lived orthodox seed, like carrot or onion. But oh baby, look what happens when you put these seeds at minus 5 degrees. It's a disaster. They died really fast. Minus 18 is better, but still relatively rapid. The seeds do okay when stored in liquid nitrogen. Even still, we see signs of deterioration in a 5-year period, and we'll have to work on the cause and try to fix it. So I've mentioned engineered components that have to function together over a broad temperature range. You should have a natural feel for this with water because water freezes at 0 degrees, and this normally causes a lethal restructuring of everything else in the cell. That's why we call it a killing frost. Dried papaya don't have enough water to freeze, so we are looking for something different. You may have noticed that a lot of the intermediate seeds are oily, so it's likely that we'll be chasing the behavior of lipids. Differential scanning calorimetry, DSC, is the preferred technique to detect thermal events like melts and crystallizations. Here, we cooled a papaya seed down to minus 150 degrees and warmed it back up and recorded an energy signal. A big peak indicates a melt, and you can see this in papaya seeds as a very clear melting signal, beginning at minus 20 degrees and peaking at about minus 5 degrees, the very temperatures that we saw the anomalies in viability. So yeah, we think that behavior of lipids are involved in the temperature response of papaya seeds We're just not sure how this is manifested. We believe we can avoid this transition that is damaging and the associated crystallization if we can just cool the seeds fast enough in liquid nitrogen. Lipids are great big molecules, so they crystallize slowly. So we think 20 to 50 degrees a minute is all that's needed, and that's completely doable. Now I'm going to present an evolving perspective about seeds that governs how they survive drying initially, why they can survive longer once they have dried, and how change eventually occurs and leads to death. It starts with a traffic jam. But instead of cars on the highway, this is a traffic jam of molecules in which they get so compressed because the solvent water is removed that they impede the movement of their neighbors. The little molecules, the motorcycles and foot traffic, are freer to move in a traffic jam. They are slower when the traffic is flowing, but in the traffic jam, they move relatively faster. A traffic jam can be frustrating, but the low speed protects molecules from actual damage. That's what we think seeds are doing. During maturation on the mother plant, they accumulate dry matter and stuff their cells. This sets up the scaffolding for the structure that comes when the vasculature between the embryo and the mother is severed and the water supply gets shut off. Eventually, enough water is removed and the molecules can't freely move and the solid matrix forms. This gives it structure. Remember the difference between solids and liquids that you learned in school? Liquids take the form of their container, but solids maintain their shape, at least for a while. But no material is perfectly stable, especially not biological materials that are always susceptible to damage by the molecules that can move. For example, the oxygen moving through the solid matrix, like the foot traffic or the motorcycles moving in a traffic jam, or interactions among neighboring molecules, like little fender benders that occur as traffic slowly creeps forward. Here I present some examples from the Archivist Chamber of Horrors that you probably encounter routinely. Old rubber bands lose their elasticity and snap. Paper browns and bears indentations where there was pressure. Ooh, and the films start to bond together so that the surfaces are hard to separate. In our dried system, the formation of a solid, some people call it a glass, and that's where the vitrification term comes in cryobiology, is a product of the storage environment. Here's a simplified view of the world, which considers biology in the context of water availability, the x-axis, and temperature, the y-axis. Extreme dryness, no water, is on the far left, and pure water is on the far right. Water, life's primary solvent, crystallizes at 0 degrees. Ice is a solid, but it has regular structure. And we can avoid ice formation, which is lethal, by interrupting hydrogen bonds or cooling super fast. Then water eventually forms a solid with irregular structure, and this occurs at minus 134 degrees. This temperature is like the holy grail for cryobiologists. You must store below it to avoid the risk of ice formation. You can see from the blue curve that the temperature needed for water to solidify amorphously increases as the solution gets more concentrated. You already know from the colligative properties of water that adding solutes also depresses the freezing temperature of water, shown by the red curve. Most of biology occurs in the narrow conditions when water is fluid, so plentiful water and above 0 degrees. Cryobiology studies life under temperatures where water wants to freeze, so below 0 degrees. Not much is known about life at the other extreme, dry conditions. We know there are organisms that survive drying, spores and mosses, brine shrimp, tardigrades, and many species of seeds. We know that the removal of water has profound impact on these organisms' metabolism, and that they cease to show the familiar symptoms of life, even though they are still alive. The blue curve demarks the transition between different phases of the aqueous solution of the cytoplasm. When it is fluid, think viscous nail polish, and solid, think nail polish that's dry. This reversible transition between fluid and solid is called vitrification. That's when the glass forms. And the reverse process is plasticization, making the glass malleable. Just to give you context, the international standards for seed storage place seeds right in the center of this space. So now we can revisit the categories of seeds. Orthodox seeds naturally dry down and vitrify, that is, they form a solid. And once this is accomplished, you can cool the material to achieve extra stability. A recalcitrant seed doesn't survive that much drying, so we partially dry it and vitrify the cytoplasm by superfast cooling treatment. And we have to cool down to the supercritical temperature of minus 134 degrees. With the intermediate seeds, we don't want to foray too deeply into the solid state, fearing desiccation damage. Rather, linger along the safety of the solid-liquid interface until temperature is low enough that nothing bad happens, and then we can cool quickly. In conclusion, we are considering seeds and the preserved state as solid systems and using the tools provided by material science engineers to understand and predict stability. I didn't talk about the aging reactions themselves and the accumulation of damage that causes mortality. Suffice to say that the kinetics of these reactions fit well into the seeds as solids construct and allows us to add the additional component of chemical composition into the overall understanding of seed longevity. Our current goal is to predict longevity on a few initial measurements that reveal structural and kinetics of early change. This will also reveal the storage conditions that maximize longevity, and we may see more flexibility in storage standards to accommodate the diversity of material we encounter. With that, I thank you for listening and welcome your questions.